So what's the law of mass action? Anybody know? No? No? So competitive binding assays are based on non-covalent reversible binding of a ligand to a specific binding protein or a receptor, right? We know all about those. And it's governed by this reaction, right? So you have some concentration of receptor plus some concentration of ligand. It's going to exist in an equilibrium where it makes this receptor-ligand complex. The rate on is governed by that on constant, rate constant. The off is governed by the rate constant of coming off. So the association rate constant is how fast it'll bind, and the dissociation rate constant is how slow it'll bind. And then you can solve for a KD. I think your book talks about KAs, not to be confused with the KD of an acid, right? So, but pretty much everybody refers to like affinities as KDs, right? So, but the only difference is the KD is equal to like 1 over the KA and likewise. Does that make sense? Do you know how you get these equations? Like I have up there, so the change in concentration of ligand receptor complex over time is going to be equal to the K1 times the concentration of ligand in receptor minus the amount coming off is K minus 1 minus receptor. And then likewise, the rate of association is like that. So at equilibrium, the two are equal. This is 0, so then that equals that, right? And then you can solve for one side or the other. KD is the K minus 1 over the K1 equals the concentration of ligand, free ligand times the concentration of free receptor divided by the concentration of the ligand receptor complex at equilibrium. So that's why with really small KDs you have really tight binding, right? Because you have much more of this than either of these. Okay? Does that make sense? You could derive one of those, right? It'd be easy. So the dissociation constant, you can also think of it as the free ligand concentration at which 50% of the receptor is occupied. Where have we heard that before? You guys probably never have, huh? Remember Michaelis-Menten? So we're not talking necessarily about KDs, but what's the constant in the Michaelis-Menten equation? For enzymes, do you remember? It's another K. KI, I think, or something like that. Is that right? I can't remember. Anyways, that if you look at the Michaelis-Menten equation, or you can just look at this. So LR, when it's half, like the total amount, it's going to be equal to the K, although this equation is looking at time. So that's not right either. So KD represents the affinity of ligand binding to the receptor. It's really 1 over the affinity because it's 1 over the affinity constant. So really small KDs, really tight binding. Each ligand has its own specific affinity to the receptor, and this can be used to define a new drug or confirm a new receptor, right? So endogenous binding proteins are things you need to be worried about in your sample. So if you're trying to measure some kind of analyte, there's going to be endogenous binding proteins, and you have to extract the analyte from those, right? So albumin's a good protein binding protein. That's going to bind some of the analytes you might be interested in. A lot of these competitive assays are used on small molecules, which are known as what? For immunological reactions, when you develop an antibody that binds to us. Yes, exactly. It's a hapten. So a lot of times you got to extract the small molecules from binding proteins in order to get a measure of the total amount. So these bound molecules must be extracted, right? And that's just what I said. And this is the kind of behavior you're going to see with the competitive binding assay, right? So you're going to have some kind of labeled ligand. It's going to be able to bind to an antibody and form an antibody labeled ligand complex. And it'll compete with an unlabeled ligand. The unlabeled ligand, just give me a show of hands. Who thinks the labeled ligand is going to be in your patient sample? Who thinks the unlabeled ligand's going to be in the patient sample? Right, the unlabeled ligand's in the patient sample. The labeled ligand is going to be part of the assay. And what's the purpose of the label? Can anybody tell me? What do you think the purpose of the label is? Yeah, to make it easily identifiable. What kind of labels do you think there could be?
Different types.
Yeah, you could have fluorophores, enzymatic labels, all kinds of different things, biotin, antibodies. So those are the difference. And this is the kind of curve you're going to see. The percent labeled ligand bound is going to decrease as you increase the concentration of the unlabeled ligand. So as your patient sample increases in concentration of the ligand that you're trying to measure, that increased concentration of the unlabeled ligand is going to displace the labeled ligand from the antibody or whatever you're using to capture that. And you'll decrease the amount of labeled ligand that's going to be bound to that antibody. Okay? And that's the whole basis of these competitive assays. So there's two different types of formats. We talked about this a little last time, right? Heterogeneous assays are probably the kind you're more familiar with, such as an ELISA, enzyme immunoassay of some sorts. So these are techniques that are going to require the separation of a receptor-bound ligand from the free labeled ligand. Okay, so that's with an ELISA. You label antibodies on inside the wells. Then you have to do wash steps to get rid of any free labeled ligand or anything away from it. And then you're going to probe it with an antibody. You wash that again. So there's a lot of steps that you have to go through to separate those two and clean them up so you can get an idea of how much of the unlabeled ligand is there. Homogeneous assays, however, do not require any separation. The activity of the label is modulated when it's bound. And we'll see some of the examples of that and how that works. So remember the difference between homogeneous, doesn't require separation, and heterogeneous requires separation, right? So radioisotopes, these are the labels. We already talked about some. So radioisotopes you can use, those are pretty much only going to be heterogeneous assays. Enzymes, so antibody ligand binding region they can modify the activity of an enzyme. Substrates, fluorophores, luminogens, electrochemoluminescent particles, or microparticles. So sensitivity, what is the sensitivity? Any gotten ideas? Like a sensitivity in an assay? It's basically a function of the affinity of the binding of the protein for its ligand, right? So if you have a really sensitive assay, you're going to have high affinity, right? Because you'll be able to differentiate small differences in concentrations. Ideally, the labeled and unlabeled ligands are going to have the same affinity for the binding protein, but that's not always going to be true, right? Because don't you think having a big label on some small hapten is going to affect its binding affinity a bit? So, but ideally, in the ideal world, they're going to have equal affinity. Detection limits is dependent upon the detectability of the labeled species, and that's also going to do how you're detecting it, right? So if you have enzymatic labels are pretty sensitive. Radioisotopes are probably the most sensitive, right? Because it's going to emit a constant amount of energy from that radioisotope, and it's easy to detect and really sensitive. In fact, you want very little radioisotope there, otherwise it'll be too noisy and you won't be able to detect it. Enzymes can be really sensitive because you're actually doing an enzymatic reaction that's going to amplify any signal that you have. Substrates, if you have it labeled with a substrate, you're using some kind of enzyme to break down that substrate into a color of some sort. So that's sensitive also. Fluorophores are going to be less sensitive because you're not amplifying any kind of signal. I guess the substrates, you're not going to really amplify any signal like you would if you're just using a labeled enzyme. Luminogen's pretty sensitive. That's because that's light produced due to some chemical reaction, right? Do you guys understand that? Luminogen, generating light. Electrochemoluminescence are highly sensitive also. You generate light once you put an electrical charge to get an electrical reaction or a redox reaction to go forth. Microparticles might be not as sensitive either. So assay detection limits is going to be the lowest concentration of ligand that can be accurately and precisely distinguished from zero. So similar to the other, we went over that before, right? Cross-reactivity. So that's the specificity is the ability of a binding protein to bind only the ligand in contrast to other substances that might be present. Percent cross-reactivity is going to be equal to the concentration of the ligand at 50% binding divided by the concentration of the cross-reactant at 50% binding. So basically the KD of the ligand divided by the KD of the cross-reactant, right? And this is an example of a common cross-reactant you're going to see. It's theophylline is one of the drugs you have to monitor, the blood levels. It's often given, you know, for COPD, I suppose, and it's actually a breakdown product of caffeine. So if you drink your coffee, that caffeine, some of it gets converted into theophylline, so you'll have theophylline in your blood when you're drinking caffeine. But the levels are very small. But the caffeine in your blood can interfere with assaying of theophylline. So when you're giving theophylline as a drug, they give a much higher dose than you would produce if you were just drinking caffeine. So the structures are very similar, so the caffeine's typically going to be able to bind to the same antibody that the theophylline is going to be binding to. So it will interfere, and this figure shows you the amount of the cross-reactivity that you can get from the assay. So the percent of labeled ligand bound, and the label is going to be some kind of labeled theophylline. And as you increase the ligand, this shows you the displacement you lose. And of course, theophylline has a higher affinity for the antibody than the caffeine would, so it shifts this curve to the right. You need more caffeine there to get the same amount of percent displacement of the labeled ligand. Does that make sense? And this is another figure from your book showing you almost the same thing, right? And another one. So ELISAs, enzymes used for ELISAs have very high specific activity, and they're going to be easily coupled to the ligand, easily V-coupled to the ligand, whatever that means. Is V and C close together? Yes. And they have to be stable also. So absent in the fluids or tissues, that's important, right? Because you don't want to, you'll get the same enzymatic activity from fluid or tissue contamination, and you have to retain that activity. And this is the overall setup for a competitive ELISA. So in this example, you have antibody bound to the inside of a well. You add labeled ligand or free ligand from the patient sample, and they're going to compete for the antibody binding sites that's on the well. You do a wash, and then you're going to add substrates after your wash. And the amount of reaction you get from that substrate is going to be based on the amount of labeled ligand there that's displaced by the free ligand. So you'll get a curve similar to this, right? A logarithmic curve where you lose displace. You're going to occupy binding sites with unlabeled ligand as those sites increase, and you'll decrease the amount of product produced logarithmically based on the amount of free ligand you have rather than labeled ligand. Another assay type format, instead of labeling in this case, you labeled the ligand with an enzyme. Instead of doing that, in this competitive ELISA, you've labeled the well with the ligand. So in this case, the labeled ligand is the ligand that's covalently attached to your well. Then you incubate antibody that's labeled to some enzyme, an enzyme labeled antibody, plus the patient sample's free ligand. And the antibody and the free ligand will compete for binding. Then you do a wash, and the amount of antibody that's left on the well is related to the amount of ligand that was competing for the enzyme. Does that make sense? So, and again, your signal will go down with the amount of free ligand you have present in the patient sample. So these are figures from your book again. So this is the capture of biotinylated antipeptide antibody as a function of biotin density in an indirect capture assay. I don't know. Stupid oak pollen. This is the reaction I always like. So this is one of the homogeneous reactions. Those ELISAs, they're all heterogeneous because you have to do all those washing steps to separate free and unbound ligands. In this one, you don't have any separation you have to do. And this is the basis for those urinalysis tests for like drugs and such that they do. They're all called EMIT, or enzyme multiplied immunoassay technique. So this one you gotta think about a little, because for the competitive ELISAs, as your free ligand increases, your signal that you're measuring decreases. For an EMIT assay, as your free ligand increases, your signal increases. Now why is that? Can anybody look at this figure and get an idea? If you examine the figure, this shows you how it works, but I don't know how well it is at explaining it. But let's see if you can figure it out. I'll explain it or tell you if you're right or wrong. What do you think? So you see a ligand there that's labeled to some big thing, and you have an S. What do you think the S stands for? And the P is product. So you have this ligand that's labeled to, what do you think that big thing is with the hash marks in it? It's supposed to represent. Well, I guess it tells you here, right? Enzyme labeled ligand. So that's an enzyme labeled ligand. Okay, and that'll produce some sort of product. And the product is what you're measuring. So in the mixture, you have your sample that has the free ligand, right? And you have antibodies there too. Those aren't in your patient sample, but they're part of the assay. So without the presence of the free ligand, the antibodies are going to bind to the labeled ligand that's labeled to the enzyme, and it blocks the enzymatic activity. So you won't get any product formed if you don't have any free ligand there to bind up the antibody. So the idea of the assay is that any ligand that's in the patient sample will bind the antibody, not allowing the antibody to block the enzymatic reaction, and you'll get some sort of product formed that's related to the concentration of free ligand you have around. Does that make sense? So that's why those P tests, you get that color right away, because if you don't produce a band there, all your antibody, right, is blocking the enzyme, because you don't have any free ligand around to capture those antibodies and not allow them to block that enzymatic reaction. So that's one of the examples of a homogeneous reaction. And this is an example of data from one of those. Inhibition of activity of gentamicin glucose 6-phosphate dehydrogenase conjugated to a monoclonal antibody to gentamicin. So as you increase the concentration of gentamicin antibody, decreases the activity. So this is the dose response curve for the homogeneous gentamicin enzyme immunoassay. So this is the emit assay. So you have, as you increase the amount of free gentamicin, it increases the activities. So the other one I like is this, polarized, or it's what's it called? The fluorescence polarization immunoassay, FPIA. So what's happening here, you think? You got a ligand that's labeled to a fluorophore, and then you have an antibody in the mix too. So do you guys remember, didn't I talk about this for the measurements using light chapter? Polarization assays. So these molecules, all of these molecules are going to spin in solution, right? Which ones do you think spin, just normal Brownian motion, which ones would spin faster and which ones would spin slower? So, well, the ligands are small, even the labeled ligand. This will spin the fastest, right? But there's no way you're going to measure how fast that's spinning without NMR or something like that. The fluorescent labeled ligand spins fast, and the antibody spins the slowest, right? Because it's the biggest. So then this will spin almost as slow, a little bit slower than just the free antibody, but it's probably going to be insignificant. But this is what you're looking for, right? The fluorescent labeled ligand. And this would spin probably the slowest out of those. But the only thing you're going to be looking at spin speeds, because you're looking at the fluorescence, is either going to be the labeled ligand that's bound to the antibody or the free ligand that's fluorescent. Both of those are going to have a spin. The free ligand that's fluorescent spins faster. So what you do, if you polarize the light that you excite with, and then you polarize the light that's coming off of that also, this light gets depolarized really fast because it's spinning so fast. Whereas this one spins a lot slower, so the light you're going to see is the polarized light. It sticks around a lot longer. It's called an anisotropic decay, I guess. So you have much slower anisotropic decay with the polarized, with the ligand, fluorescent labeled ligand bound to an antibody, because it's such a big one. It spins the slowest. So as you increase the amount of free ligand, you're going to decrease the amount of labeled ligand that's bound to an antibody, and it'll spin a lot faster and depolarize faster, so you'll see less fluorescent polarized light, less polarization in the light. And I put this back up here. Didn't I show you this before? From a long time ago, did I? You don't remember? So you know light's composed of electrical and magnetic waves at right angles to each other. So light waves produced by standard excitation have, they'll produce these vectors and they're not oriented at all. So you'll randomly have vectors. I like this figure best. So you'll produce the waveforms, all the vectors will be in all disordered array, unless you shine it through a polarization filter, which only is going to let certain vectors through that are oriented one way or the other. Then they're going to strike some compounds that are fluorescent. The ones that are large spin the slowest, so the light emitted from those will remain polarized. And then you can run it through a polarization filter again. And if it's oriented one way, you can detect the polarized light that comes through, or you can block it if you do the polarization 90 degrees from that, and you won't get any light through. However, for small molecules that gets excited and then it's spinning really fast, so the light it emits is depolarized. You're emitting light with every angle vector. So you'll be able to see polarization through both filters, basically, because you'll get light that's depolarized and a little bit of light that's polarized. You should see more light that's polarized coming off. But do you see how that works, kind of? Fluorescence polarization immunoassay. So the important thing to remember is that if you have the labeled ligand bound to the antibody, that's where you'll get the biggest signal. And as you increase the free ligand, you'll see less signal, because you're going to displace that fluorescent labeled antibody. Or not fluorescent fluorescent labeled ligand bound to the antibody. So, and I looked up that assay you were talking about. There's nowhere I find where they tell you how that one's done. I found people talking about cabbage. Like for measurement of pH, I guess, or something. So this is your simultaneous electrochemoluminescent competitive binding assay, where they have a biotin estradiol and then an estradiol that's labeled to a rubidium BPY3+2, which will react down here. So basically, you put that in the mix, right? These three, the estradiol is what you're trying to measure. The estradiol, the labeled estradiol is part of your assay, and so is the biotinylated antibody. So that biotin with the antibody is just a biotinylated antibody. Then we discussed a little bit last time, what does biotin bind to with high affinity? Avidin. So you mix these together till you get your complexes. You have biotin antibody labeled to estradiol, and then you have the biotin labeled to the labeled estradiol, and this is the what you're competing against this with. Then you're going to add streptavidin, and the streptavidin is bound to this big magnetic bead, okay? And that'll pull down any of these biotinylated antibodies that are going to contain the labeled estradiol versus the unlabeled estradiol. But only the labeled estradiol will generate a redox reaction at the electrode, and you'll see current from that. And the current that's generated is inversely proportional to the amount of free estradiol that's in your reaction mixture. Does that make sense? So that one's a bit more complicated, but it's really not that complicated. So, and then this is the oxidation reduction reaction that takes place at the electrode. You get oxidation, which gives up a free electron, and then you'll be able to measure that activity, or it emits light rather. So, depending on which type of reaction you're doing. And this is another example of a similar reaction with that. So this one, so principle, the homogeneous luminescent oxygen channeling amino acid, the Loki method. So you have a photosensitized dye, and the sensitizer particles generates a singlet oxygen that's then going to react with the dyes and the chemoluminescer particles paired by antibody ligand binding. So the light plus the sensitizer is going to yield a singlet oxygen, which is going to react with the chemoluminescent, which will emit light, then fluorescence. So you have a biotin labeled ligand and a ligand that'll compete for binding sites on the chemoluminescer, and then streptavidin labeled to the what do you call it? The sensitizer, which is required to be there. So that's labeled with streptavidin, so it'll only bind to the biotin labeled ligand. So the more you have biotin labeled ligand, the less free ligand you have in that solution. And the more free ligand you have, the less biotin labeled ligand you have, which the less sensitizer you'll have there, which means the less fluorescence you'll get when you expose it to light. Does that make sense? Because you'll slow this reaction down, because these two won't be close enough together. And that's it for the competitive binding chapter, I guess. So I had planned a short lecture today because I thought we were going to do that. Eliza, are you guys mad at me?
No.
Yes, you are.
We're really mad.
But you guys can, I guess, use this time to work on the exams.